All right. And now, to conclude this year's symposium, we have our two recipients of MSI's Distinguished Achievement Award. Now, earlier this year, as members of the steering committee and leadership were thinking about who do we give this to? There were really a lot of deserving scholars in our community. We have this extraordinary community of scientists who do incredible work, but also community service, right? Really engaging across disciplines and across institutions. Excuse my allergies here, but there are two individuals who stood out among this very distinguished crowd, and those two, Drs. Colleen Cavanaugh and Dr. Roberto Coulter, are the recipients of this year's award. Now, I have the great pleasure of introducing Dr. Colleen Cavanaugh, who has been my colleague, mentor, copy editor, BioLab's upstairs neighbor, advocate, and friend since I have been here. In fact, my first month in Cambridge, after having moved from sunny California, was spent in her home, which she kindly lent to my wife, Christy, and I as we found a place to live. And in exchange for a few household repairs, we ended up staying there for about a month and really getting to know Colleen and Phil. And I've really had this great pleasure of getting to know Colleen over the years. Now, I first met Colleen at a Symbioses conference. It was quite a while ago. And while I don't actually remember exactly when I first met Colleen, I certainly remember meeting Colleen. How many of you know what I mean? Right? As they say on social media, if you know, you know. I first met, it was definitely a Symbiosis conference, and Colleen, you were holding court and talking about some of your favorite subjects. And what I recall is carbon fixation and other topics, including but not limited to PCR bias, symbioses, sulfur oxidation, mold, real estate, and of course, pandas, which, by the way, for the record, are not bears. Just don't get yourself in trouble with Colleen. But so are they now? I don't know what the hell they are. You know what they, to ask Colleen about pandas. Now, what I witnessed at that first meeting, though, was Colleen at her very best. She is really a brilliant scientist with an impressive ability to identify patterns and processes across scales of biological organization. This is true. That has allowed her to make discoveries that her colleagues at times refuse to believe. From her groundbreaking discovery of the first chemoautotrophic symbioses and to her work on the activity and isotopic fingerprints of rubisco, and to her latest work on studying oral biofilms, Colleen and her lab members have always pushed the envelope to help us advance our understanding of how microbes make a living in this world and upon us. Now, Colleen hails from Detroit, Michigan. She attended the University of Michigan, where she majored in ecology. But her sophomore year, Colleen participated in a research project on the mating habits of horseshoe crabs here in Massachusetts. It was there that she fell in love with the interdisciplinary nature of marine science. She delighted in talking with biologists, geologists, and chemists who worked side by side, rubbed elbows in the field, drank beers together to better understand the ocean and its denizens. Now, when Colleen finished earning her undergraduate degree, she moved to Woods Hole to work at the Marine Biological Laboratory, where over the next 2 years, she fell in love with creatures that impressed her for their ability to live anywhere, namely the microbes. She came to Harvard as a graduate student, earned a master's in 1981, a PhD in 1985, and then joined the Society of Fellows as a junior fellow from 1986 to 1989. That led to an assistant professorship here, an associate professor in 1993, and a tenured full professorship in 1995. Now, of course, as you all know, Colleen has been doing amazing work in that time. And then in the early 2000s, around 2003, she and Dr. Coulter were the first to be the co-directors of the Microbial Sciences Initiative, and were primarily responsible for creating the vibrant community that you see here today. Now, Colleen has authored or co-authored about 120 or more publications. It was just hard to count them all. Participated in a dozen or so deep sea research expeditions, received dozens of awards and accolades, including induction as a fellow into the American Association for the Advancement of Science. And her work has reshaped our understanding of microbe-animal associations, carbon fixation, and her previous and ongoing leadership in our community is exemplary. This is why she has earned the MSI Distinguished Achievement Award. And it's been a real honor to know her as a mentor, colleague, and friend. And so without any further ado, let's welcome Dr. Colleen Cavanaugh.
Well, thank you very much. And thank you, Peter and Mike and the steering committee and leadership for this very honored, I'm not honored, I am honored to be part of this award and sharing it with Roberto. Interestingly, perhaps, I gave a talk our first symposium 21 years ago. So this is my 20 years later. But I want to say how honored I am to have been co-director with Roberto and to see that MSI is still thriving, that it is bringing together, as I always called them, as we organize things, the MMs and the EEEs and everything in between. That's molecular biology, molecular, micro, medical micro, ecology, evolution, and engineering. And this you've seen from the talks today, and when you look around you and meet people, that this has really worked to bring people from all across Harvard, as well as the surrounding areas and from outside. So today, what I'm going to do is a bit of a retrospective. I'm going to focus on chemosynthetic symbioses. Figured since I talked about it 20 years ago, it was time to update. And the areas I'll talk about is the background on the actual discovery, just as it paralleled life in microbiology or microbial ecology pre-omics and pre-molecular micro. Then I want to talk about, Nicole gave a great talk. I'm going to be talking about transmission strategies in these chemosynthetic symbioses and how varied they are. And then if time, I'll talk about the case of the missing enzyme. And I can summarize that if I need to. Okay, so this, I know I'm preaching to the crowd, but I want to, in the context of symbiosis, have you realize just how many bacteria you experience. Whether you're drinking water, showering, breathing, playing in the dirt, and that's a low estimate for the atmosphere and air. There are microbes everywhere. They've been here, as we learn, repeatedly since first fossil record is about 3.5 billion years. And their metabolic capabilities and strategies have co-evolved with the geochemistry and the biogeochemistry of the Earth over those 3.5 billion years. So it's no surprise that microbial symbioses are ubiquitous in nature. And this is both because you're immersed as every organism, whether you're a bacterium, protist, virus, et cetera, you're born or you're evolved into a microbial milieu. And from the point of view, especially of eukaryotes, who have a very limited metabolic suite, basically fermentation and aerobic respiration only courtesy of their mitochondria, and then photosynthesis courtesy of their chloroplast. So really fermentation. But given the broad metabolic suite of metabolic opportunities, these symbioses have formed repeatedly. As we've heard repeatedly as well, and it's part of the norm now, that the majority of microbes are unculturable. And the revolution in microbiology, so-called omics, was heralded with Carl Woese and his revolutionary, looking at the origin of not origin of life, but the ancestor of all life, and using molecular sequencing, using cultured organisms. And then the leap that Norman Pace made to be able to look at uncultured organisms in the environment. And this kind of parallels my scientific life. I actually started working for John Hobby, who developed the method for counting bacteria in the environment. And I was the one that worked on counting, getting it to work for soil and sediment. So that's where I started. Okay, that's my other soapbox, which I think I've said before. This is the EE part of me. And that is that we know that, well, everyone I think accepts that nothing makes sense except in the light of evolution. But I always want to add, except in the light of ecology and evolution, because that's the selective pressure, that's what organisms are growing up in, and it's everything from the physical to chemical to biological interactions. And so you have bacteria interactions, you have chemistry, and I always say for medical microbiome people, I'm not speaking to anyone specific here, that I always think they should have an evolutionary biologist on their one shoulder and a microbial ecologist on the other to really, embrace not just the parasite or pathogen aspect, but to look at it as an ecosystem and really a human body as a habitat. And I do want to make one other little soapbox thing, and that is there's no such thing as a bad bacterium. They are not out to get you. They are out to eat, and they've evolved eloquent ways of getting food, and sometimes you just happen to be it. So I know they're bad for us, just bad bacteria. No. Okay, history. Not as far back as I think probably Roberto's going. So deep sea hydrothermal vents, the discovery of these chemosynthetic symbioses was preceded by the discovery of hydrothermal vents and then the discovery of gutless animals within a very short period of time. And for those of you who may not have thought about vents in a while, they were discovered in '77. They're hot springs on the ocean floor associated with areas of seafloor spreading. And it's a lot of volcanism, seawater percolates in, so you get a very changed chemistry with very hot temperatures like a black smoker, but then subsurface mixing, you have kind of warm water full of chemicals that bacteria can eat, and archaea, I'm not to diss archaea, it's just too bad prokaryotes is not allowed to be used. And organisms, though, that can use hydrogen sulfide, methane, CO, single carbon compounds that metazoans cannot do, and most protists. And the initial discovery was that chemosynthesis was the base of the food chain, and that was proposed when they first smelled sulfide in some of the water samples. And this, of course, revolutionized our perception of life in the deep sea. There's these flourishing ecosystems, giant tube worms, clams, mussels, shrimp, snails, it's enormous. And the base of the food chain for that is 2.5, 3.5 kilometers depth. So it's not light-based, it's chemical-based. And this changed our view of symbiosis as well, of chemosynthetic. So the first gutless animal were giant tube worms, Riftia pachyptala, which were unusual. It ends up being annelids, but they have no mouth and no gut. And as a first-year graduate student, when I was in a class, Meredith Jones, we had a series of talks on the vents, and he talked about these worms, no mouth, no gut. It was a 1 micron by 1 micron talk through a 2-meter-long worm. Unbelievably, I was awake. When he mentioned that he found elemental sulfur crystals in the tissue that filled the coelomic cavity. And this was the clue that suggested that there were chemosynthetic bacteria that could oxidize sulfide to intermediates like elemental sulfur and fix CO2 and feed the tube worms. And indeed, we were able to show that there are 10 to the 10th, 10 to the 11th, 10 to the 12th bacteria per gram wet weight. They're intracellular. Horace Feldbach at Scripps measured rubisco activities indicated the Calvin cycle and other sulfur enzymes. And we proposed that indeed we had a chemosynthetic symbiosis and that the bacteria, the worm, is providing the necessary compounds and the bacteria would feed the worm. There are numerous adaptations, which I won't have time to go into, but I'm happy to talk about. So shortly after this, there was a paper in Science, Gutless Bivalves. That was the title of the paper. And it turned out it was about a solomyid protobranch bivalve, of which Solomia velum lives in the Woods Hole area, and in New England mudflats. And of course, it took vents to see this symbiosis, to see the need for sulfur. And in mudflats, you could go out and smell sulfide. It's biologically produced. So looking at this animal, able to find bacteria intracellular within the gill tissues, similarly using enzyme activities to localize to identify them. The cool thing about these, they have a behavioral adaptation. They make Y-shaped burrows, and this facilitates one of the adaptations to get the sulfur. You get oxygen from above and sulfide from below. And this shows one we happen to catch in situ. Okay. So basically then, chemosynthetic symbioses are effectively like chloroplasts, only using sulfur instead of sunlight. And the host benefits from a non-metazoan energy and carbon source, But the key is that the symbionts are bridging an oxic-anoxic interface. And so we and researchers around the world started looking everywhere and found them in all of the major macrobes at hydrothermal vents, the macrofauna, macrobes, harbor chemosynthetic symbionts, the bivalves in their gills, oligochaetes and nematodes. The nematode has them as epibionts, and they really are extremely widespread. And while all this was going on, so we're using microscopy, enzymology, but I couldn't culture the symbionts. Now, let me tell you, for the real microbiologists in my life at the time, and hopefully many of your worldview has changed, I was dead to them. Holger Janisch, Pete Greenberg, you can't culture it. What are you talking about? So, fortunately, Norman Pace gave a talk in Woods Hole and talked about using the ribosomal RNA, at that time it was 5S ribosomal RNA, for octopus springs, to get at, he made the leap that you could just extract the RNA and sequence it. And I pounced down afterwards, arranged to get him Solomaya, the Riftia and clams, and extracting the 5S RNA, I'm just showing this for historical reasons, and actually directly sequencing it, they were able to, show that, I guess I have to do this. The symbionts fell within, if you can see them. Can you see this? No, not really. Phyliptigina, Solomaya, and then the tube worm. And they fell within what were called at that time the purple photosynthetic bacteria, which we know is proteobacteria. And this is actually, Norman, the first paper using molecular sequencing to identify uncultured bacteria from a habitat. They went on then to publish the octopus springs on using 5S, and then moved to 16S using reverse transcriptase, and then PCR came on the scene, and we all know what's going on. Omics, omics, omics. So that was the beginning. As I mentioned, they're ubiquitous in nature. Numerous habitats, shallow to deep, mudflats, sunken whales. There are 27 animal families, paper by, what's her first name? Sogan and Nicole Dublier. 7 phyla, 3 protist groups, and 1 archaeum has actually got sulfur bacteria on the outside of it. The energy sources are sulfur, hydrogen, and methane, carbon, CO2, and methane. And then for the 16S, they tend to be a single phylotype, but there's many variants now, but this is kind of the general. They're mostly gamma and alpha proteobacteria, and then Campylobacter tends to be the epibiotic ones at the bottom. There's a wide range of host-symbiont interactions figured out by sequencing, and that includes things like type 2 secretion, virulence factors, and flagella, suggesting that we're trying to figure out where are they coming from, transmission-wise. Okay, transmission, dancing in the dark. This is the question, as Nicole so nicely talked about, thinking about transmission, vertical or horizontal. The issues have implications not only for genome evolution, for the host and symbiont evolution, but also for microbial diversity in the environment. With vertical transmission, bacteria pass directly from mother to progeny. And if they're intracellular, that's where you have this bottleneck and you end up with a very limited population, clonal symbiont populations, and then you tend to have gene loss and get smaller genomes. With horizontal, you acquire the symbiont from the environment, so that gives you a chance to get various variant strains that may be more adapted to that particular environment, and also to allow for recombination. With free-living bacteria, it also seeds the environment, so you're actually impacting the local bacterial diversity. Now, mixed transmission was something that was a little bit in the literature, but as you'll see with ours, it has features of both vertical and horizontal, and now appears to be much more widespread than thought initially. So the first was for the giant clam. It was the first genome to be sequenced. This was in collaboration with JGI at the time when sequencing was not easy. And it turned out it was a very small genome relative, for reference, E. coli is 4 million base pairs. So it was small, but we were able to close it. It had all key genes for chemosynthesis. And while it looked like Craig Carey found the bacteria in the follicles and eggs, so it indicated that it was vertically transmitted, But there was a little bit of evidence from a couple of other viscocamaions that there may be some recombination. But generally, we're figuring vertical. You're in the deep sea, you want to have your bacterium with you. Well, tube worms. Wouldn't you think if you're mouthless and gutless as an adult that you would want your bacteria with you and they would be vertically transmitted? But all of the evidence came back, a lot of it was negative. There's no bacteria there. But the evidence for horizontal was that the 16S phylotypes of the worms matched habitat. There'd be different species of the vesimentifrans, and it matched habitat rather than the actual phylogeny of the host. The other was the genome was quite large, 3.2 million base pairs, full metabolic gene suite, and they had a number of free-living genes. And this, by the way, is not all my work. I've tried to put people's names on here. So it's been a long group. In fact, I should note, Peter Gergis's group, and a number of whom students and postdocs and colleagues are here, is one of the pioneers in the physiology and biology of these, as well as genomics. So we'll hear from more of you next time. So the question was, okay, if they're free living, then they have to be out there. This is the only exploratory grant I ever got. We basically put down this evidence and said, we want to go look for them. And we got funded. And it was by a NERP, National Undersea Research Program, through the University of Alaska, NOAA, which I commend them, but they let us go. And indeed, we put out bacteria traps, and we're able to show that we found the symbionts based on fish and probes, that they were both in biofilms and in the seawater. And then sequencing using, at that time, 16S and ITS showed that there was a dominant genotype in most tube worms, but there could be at least two, and then there were some variants. And in the seawater, there were a number of different variants. And there's been considerable work now on the genome and looking at these in the environment. And I should say it, and the most symbionts are right at the site of the worms, and they are, it's been shown by other groups that they actually release symbionts when they die. So it makes sense, you're seeding the environment. Symbiont acquisition, how do they get them? Okay, they're mouthless and gutless, it turns out only as adults. They have the larvae have mouth and gut, but is that how they get their symbionts? No. Work by Monica Bright and her student showed that the bacteria in a larva, notice that's only, I think, 4 millimeters in length, they come in through the body wall, affect the development of that trophosome tissue, and at some point apoptosis happens and it stops. And so it's a completely unexpected. And again, remember, these are tube worms. They have to spawn at the same time. They're in the dark, eggs and sperm. They have to find each other. They have to find a crevice that isn't going to be grazed by a snail or something that's a grazer, and they have to find their symbionts. So it seems absurd to me, but it's evolution. This is how it happened. So how about mixed? Mixed transmission. So Solomaya, we had a lot of evidence for it being vertically transmitted, finding bacteria in the oocytes, and then bacteria able to actually spawn them in the lab in very young juveniles, 11 or 64 days, within the gill buds. And so we had the expectation, as we did with even Riftia, that they were going to be vertically transmitted. But then, when we got the genome sequenced and analyzed it, it's a relatively large genome. It has virtually every full metabolic pathway. And so the question looked like it was horizontal. So Shelby Russell, this might be what Nicole was referring to on mixed transmission, a graduate student in my lab, went out to test this at the population level, because with the two, it was like, is it vertical, is it horizontal? Because mixed transmission was a little bit in the literature, but not really. It was really, kind of the dogma is vertical or horizontal. But what she did was she had 5 different sites, you can see the dots in color, 7 to 21 clams per site, 61 clams total. She did metagenomic sequencing of the gills with the entire host and the symbiont, focusing on the mitochondrial, reconstructing the mitochondrial and bacterial DNA based on Solomaya velum references with very high coverage. By the way, a lot of these were from historical literature, finding the sites. I mean, 1800s, like where Solomai is. She'd get there and it'd be a subdivision, and she had to go with her shovel and her buckets and kind of wend her way through people's yards. It was a monumental effort. That's one of those things you don't think about or you don't see in the methods. What she found was geographically, for vertical evidence, they were geographically congruent. So host and symbiont within a certain geographical region, indicating inheritance through each individual. But the horizontal evidence, if you can see well enough, was a discordant genealogy between, this is where, you can see mitochondria and symbiont. You can see the cross lines. So they did not, it wasn't a straight, paired genealogy indicating horizontal, indicating suspected horizontal. She then analyzed the genomes, and there was incredible amounts of recombination, genes from other bacteria in there, and there was a very dynamic symbiont genome structure. It was different between different animals. So the key here is that with the mixed population, you really needed this population level analysis needed to actually discern whether it was vertical or it was horizontal. And this is where we're seeing the mixed transmission. Shelby was an amazing literature hound. And this was discussions we had over the course of her 5-6 years in the lab about the mixed vertical. And she did a meta-analysis of the literature for all symbioses that she could get good data on. And you can see the number, 528, 113, and looked at the transmission mode versus taxon, environmental type, and other metadata, and found that transmission fit with the taxon, which makes sense within a group, but weirdly, and that there was equal numbers of vertical, horizontal, and mixed. And that the binning by environment, this was just something we don't really quite get. Terrestrial had a lot more vertical, so that's why I'm interested, Nicole, in the bugs, because you did have symptoms of both. And then aquatic was more horizontal. So the bobtail squid is horizontal. Here I was gonna say, this is just viewing it better as a spectrum. Other clams, but aphids and other sap-feeding insects typically are thought to be vertical. We're gonna have to change that figure, Nicole, I don't know where you are. So this really had us thinking in a different way about transmission. And one of the points that she brought out in the paper is that there's likely a lot more mixed transmission because of the way that things are studied. It hasn't been looked at that extensive, comparing the actual genomes. And it makes sense because if you have that ability, as I mentioned, to adapt, to have an adapted symbiont, and there's now papers out on that from even the tube worms, from a local habitat, and for the possibility for recombination. So it makes perfect sense, and it's now, I just saw in reviewing things for this talk, that it's just mixed transmission has become like, they just call it normal now. So there's already 3 types. So, where am I? 10 minutes. Okay. The case of the missing enzyme, I can summarize this because I know you want me to. One of the crazy things about all of this sequencing is that, well, it's not crazy. The majority of the chemosynthetic symbionts fix carbon via the Calvin cycle. The tube worms also have the reverse TCA cycle, which Jesse and Peter's lab has studied. But the majority have the Calvin cycle. And we can find it in virtually the entire cycle in the various genomes. But every sequenced symbiont to date is lacking one of the key enzymes, the FBPase, fructose 1,6-bisphosphotase. And it's in the regenerative phase. So it takes fructose 1,6-bisphosphate to fructose 6 and cetoheptalose 1,7 to cetoheptalose 7. And so the cycle is incomplete. And when we first found this, Irene Newton and then others who found this too, hypothesized that a reversible pyrophosphate-dependent phosphofructokinase could serve as the enzyme instead of the FBPase. And I'm going to blitz through this. Oleg tackled this, Dmytrenko, and the question was, could it work? Now, we can't culture the bacteria. We can't, there's no genetics. So this was all done either with cloning. So is the PFP expressed? Yes, it is. Transcriptomics, and it was expressed at the same level as other, the Calvin cycle enzymes. Activity? Yes, measured in very high activities in the gill tissue of the clam where the bacteria are. And then could it perform the biochemical function? He put recombinant Solomia velum into E. coli and was able to show that it worked. So that's summarizing years of work. So he wanted to show it experimentally, not just this genomic stuff. So he found through the literature and a colleague, Allochromatium venosum, which is an anoxygenic photosynthetic bacterium, closely related to the symbionts. It's a gamma proteo, and it has both FPP and PPPFK, and is genetically tractable. And one of the coolest things is it can grow photoautotrophically, but it can also grow chemoheterotrophically, photoheterotrophically. Anyway, both ways. So you can grow it much easier than growing an anaerobe. But to grow the anaerobe, he created this anaerobic bioreactor, in part buying parts off eBay. He had it was all automated, pH regulated, measured, had sulfide electrode, and then they had the OD being measured automatically. You'll have to believe me, I didn't mention the knockout. So he made double knockout mutants, and you have to believe me that he tested everything backwards and forwards. I am a control freak, so they all checked out, and we're going to jump right to the final one. So when he tested this bacterium with or without the PFK, the PPI PFK or the FBPase, it grew autotrophically, photoautotrophically with both. You can see with the normal enzyme, which is FBPase, the knockout for the PPI, grew faster. They had a longer lag time. This is a typical change from heterotroph to autotroph lag time, but the PPI PFK positive took longer, and we speculate that might be. I'm too loud. What's that?
It's easier for you.
Oh.
So you can talk
Okay. That it could either be just to get the proteins, the enzymes transcribed, or you have to get the PPI, the pyrophosphate concentration, to be very low in cells because it inhibits the reverse reaction. So he was able to show this. One speculative question is whether this is an energy-saving routine for these chemosynthetic symbionts, because you can get additional energy making ATP. There are other enzymes, but this particular one, the proton pumping PPPase, is also in all of the chemosynthetic symbionts. And so that's what we propose, is that there's the Calvin cycle 1.0 and then Calvin cycle 2.0 using the pyrophosphate PFK as a reversible enzyme. So that was it in a blitz. Clearly, the point about ecology in particular is that it took the vents, it took the lack of sunlight to be able to focus on sulfur as a potential energy source for fueling an ecosystem. And there are so many bacteria that eat weird things that I'm sure that this is just a wealth of area for discovery. The dancing in the dark, we were very surprised, expected totally vertical in the ocean, and certainly in the deep sea, but it led to the insights into this mixed transmission. And then the missing enzyme, well, we think we've solved it, so there we go. And to acknowledge my hopefully mutual, my symbionts in a symbiosis that's hopefully been mutualistic. Let's see, what else was I gonna do? Oh, I wanna mention that for those of you who are interested, I have segued into the terrestrial realm, as well as the air with the oral microbiome Peter mentioned, and eating is here, my student who's working with, collaborating with Tina Werner on the ancient microbiome, oral microbiome, and one of the CPR bacteria. And the other is the birds, the air. That's Darwin's finches and more oral. And I have to do this. I'm in the Department of Organismic and Evolutionary Biology, and Ed Wilson, who passed away a couple years ago, for those of you who have not read his autobiography, The Naturalist, the last paragraph is, if I could do it all over again and relive my vision in the 21st century, I would become a microbial ecologist. And he was such a champion of microbial diversity in the later part, he's been conservation and biodiversity. So I just love that. So I will close there. Thank you.
First awardee received this stromatolite. So this one is yours. I can hang on to it for you if you want, or I can give.
No, I don't.
Okay.
Thank you.
But there is time for questions, so please.
Oh my goodness. I need Tanya to interpret. Any questions? Oh, yes.
Thank you. That was a great talk. So the missing enzyme, has that been lost multiple times, or is that like a single origin of a switch to a new Calvin cycle?
We are in the process of doing an ancestral reconstruction, which was done with fewer genomes ahead of time. But so far, it's not only missing in the chemosynthetic symbionts, it's missing, it turns out, in a number of methylotrophs and methanotropes as well. And it would thought to be functional more in anaerobic glycolysis or gluconeogenesis, goes both ways. But now in the C1 organisms, they're postulating that there is some sort of energy savings, but I have not been able to discern. It's been a little sketchy what they're getting at. So I think it's going to be more widespread, but so far that's the only place, and we keep searching. I've stunned everyone.
Wonderful, Colleen. Thank you so much for that. And I fear I'm oversimplifying, so I want to just tell you what I took away and then give you the opportunity to straighten this all out. So I thought that was super cool, the transmission patterns that you were finding. And it struck me that it was like, okay, if you live in the water and you need a symbiont, you're going to go horizontal. And if you live on terrestrial land, you're going to maybe be mixed or vertical. What do you say about that?
Mixed or vertical? Well, in terms of why?
Or just, do we think we're coming to some principle about symbiont transmission, that it's like when you're in aquatic environments, it's going to probably be a horizontal symbiont relationship?
Well, yeah. The rationale in thinking about it is that water is much denser than air, so that when you have a release of these bacteria, then they are contained, because we had orders of magnitude going out from right within two worms out to 100 meters off. We still detected them out that far, but they're concentrated. And if you think about it with rhizobia and legumes, the bacteria are released from the bacteroids within the area of the root, and that's horizontal transmission. Then they're picked up by other plants, but it's contained by the soil matrix. And so that's one of the things. And Shelby cites quite a bit of literature showing the number of symbionts are transmitted via, like they use the plant sap to actually move them, so giving it a fluid mechanism. So that's one of the lines of thinking. I mean, mixed makes sense from it does and it doesn't. There's the argument, they're there. I mean, for the deep sea, it seems crazy to me, but.
Great talk. Based on what you just said, do you think the environment that the organism is living in, like the density of the water, the density of the soil, the density of the air, is the contributing factor that makes an organism horizontal, vertical, or mixed?
Well, that's our speculation.
Yeah.
I mean, because we learned very strongly with COVID how fast you spread. And so if you're in proximity, the thing is with sneezing and coughing, you actually can propel saliva droplets up to 100 miles per hour. So that's another mechanism. But for most of these insect symbionts that are inside specialized tissues or something, the idea of even the vertical transmission, a lot of it is just what Nicole said. They actually deposit pods of bacteria that when the larvae hatch, they're eating them, they're right there. So I think it's really an issue of containment. And in the water, it's still a very dynamic environment, but at least we showed, we were able to measure it. Similar with the bobtail squid, the bioluminescent bacteria. It eviscerates 95% of its bioluminescent bacteria in the morning. And Ned Ruby and his student, basically, I always picture them in a rowboat, I don't know why. They went out in Kaneohe Bay and took samples all the way out, and the same order of magnitude going down. So right around where the squid lived were a lot more of the bacteria. And of course, they're being seeded.
Interesting, thank you.
Yep.
Very nice talk. I'm just curious, kind of on a general level, you mentioned at some point that a lot of predecessors in the field felt that anaerobes were unculturable. Why do you think that is?
Oh, sorry. I didn't mean to say that. Just the reason we know that the majority of bacteria cannot be cultured are that we can count them directly now. So before, everything relied on colony-forming units, but we now know that there are 10 to the 6 bacteria in virtually every water body. It goes up and down, and 10 to the 9th per gram of soil or sediment. But if you go out and try to culture something from soil or from the gut, you get X number versus what you can actually directly count. So it really took that understanding just how many, and they're really tiny, the typical environmental bacteria, not in the gut, but so you couldn't see them with a light microscope either. It had to use this epifluorescent technique, staining the DNA so that you could recognize them.
Thank you, Colleen.
Oh, okay. Oh, this guy's here.
One more. Can we take one more? Let's do one more, Colleen.
I'm not sure. I thought somebody was back there. Oh, okay.
My question is about the mixed transmission. So my understanding, which might be wrong, is that these like gutless organisms have like only a handful of taxa that are really like supporting them. So in the like mixed transmission, is like the vertical and the horizontal taxa similar, or are they like grabbing from different pots?
Typically, in the animals that have the horizontal transmission, they have a number of variants, but it still tends to focus on a single 16S, or in the case of dual symbioses with methanotrophs and sulfur bacteria, But there are definitely variants, and it can vary with geography, or it can vary with taxa. So that's coming out now with all the metagenomics. But clearly there's been recombination, so it has to have been reasonably recent in this trajectory. Okay.
Great, last word, baby.
Last word. Yes, to Colleen, thank you very much. Let's give Colleen a round of applause for an amazing presentation.